Good morning, everyone. We will continue our discussion on the methods for the development of transgenic plants. So, the basic principle of plant transformation as mediated by the bacterium, Agrobacterium tumefaciens, is about the selection of one or more transformed cells and also about regeneration of these cells into a transgenic plant. Number one, it is about the selection of one or more transformed cells. And number two, it is about regeneration of these transformed cells into a transgenic plant. And many different types of plant tissues, which include somatic embryos, tubers, cotyledons, and fragments of leaves, stems, or even roots can be used for the co-culturing stage in which both the bacteria and the plant cells are in contact. So there is possibility for the use of different types of plant tissues including the somatic embryos, the tubers, cotyledons, and fragments of leaves, stems, or roots. So they are used or can be used for the co-culturing stage in which both the bacteria and the plant cells are in contact. And even some of the compounds, say for example the phenolic compounds such as acetosyrinzone, hydroxyacetosyrinzone, and others, they are added to the co-culture medium for the reason that to facilitate the initial steps of transformation and also to enhance the recognition, contact, and finally the transfer of the tDNA. Right. So you can have different types of plant tissues for or to be used for the co-culturing stage wherein you find the bacteria and the plant cells are in contact. And number two, phenolic compounds, for example, acetosyrinzone, hydroxyacetosyrinzone can be added to the co-culture medium so that you facilitate the initial steps of transformation and enhance the recognition, contact, and transfer of the tDNA. Right. So after the co-culturing period, the explants, what we refer to as the small pieces of the plant tissues, they are maintained in a specific medium which contain a selection agent and an antibiotic that prevents the regeneration of untransformed cells. Right. So the dosage and the duration of exposure to the selection agent that depends on the laboratory conditions as necessary. So it can be adjusted to the requirement. So what should be the dosage? What should be the duration of exposure to the selection agent? So that varies. After several successive cultures, only the transformed tissues will regenerate and grow into transgenic plants. So this is a theoretical approach. After few successive cultures, only the transformed tissues will regenerate and they grow into transgenic plant. But there is another factor which is coming into picture. We refer to this as leak occurrence. Now what is leak occurrence? Is in which the non-transformed cells can generate in the selective medium. Right? So we expect that after successive cultures only the transformed tissues will regenerate and grow into transgenic plant. But there is a possibility that non-transformed cells can regenerate in the selective medium. So this is referred to as leak occurrence. Therefore, the transgenic state of the plant, whether it is transformed truly, must be confirmed before it is taken into the greenhouse conditions. Right? Before it is taken for acclimatization to the natural environment. So the transgenic state of the plant, whether it is truly transformed, whether it is truly regenerated from the transformed cells, has to be confirmed before it is taken into the greenhouse acclimatization conditions. Right? So then genetic transformations using agrobacterium tumefaciens. So it is the most commonly used process to obtain transgenic plants for the reason that number one, it is a low cost method, and number two, it is a relatively simple method compared to any other methods that are available. Number one, it is a low cost method, and number two, a relatively simple method. And most important is this genetic transformation process result in fewer copies of the transgene and is considered more accurate than the direct method of gene transfer. Right? So this is supposed to be the most commonly used process to obtain transgenic plants for the reason that it is a low cost method and relatively simple compared to any other methods that are available. Right? And Agrobacterium tumefaciens has a very low capacity to infect monocots. So this is a limitation, an important limitation of this method. Agrobacterium tumefaciens has a very low capacity to infect monocots. So this limits the application of this transformation method to an important group of plants, say for example corn, rice, wheat, etc. because it is having a low capacity to infect monocots. So an important limitation of the application of this method to the monocots, the important ones are the corn, rice and wheat. Is that clear? So moving further, we have the next method, that is the direct methods. Here the introduction of genes into the nucleus of the target cells by direct methods is dependent on making physical and chemical changes to the plant cell walls and the membranes. So the direct method is available for the transfer of the gene of interest, but the introduction of genes into the nucleus of target cells, when the direct method is adopted, these methods are dependent on making physical and chemical changes to the plant cell wall and the membrane. Right. So the exogenous gene to be introduced into the cell nucleus is inserted into a vector. The vector is then adhered to a metal microparticle for its introduction into the cells and transformation of the recipient cell. Theoretically, direct methods may be used to transfer DNA to virtually all plant species. There should not be any barrier for the reason that the direct methods are dependent on making physical and chemical changes to the plant cell wall and membranes. So definitely it is accepted that this or these direct methods can be used to transfer the DNA to virtually all plant species. It is not so, but theoretically it is assumed that it may be used for transfer of DNA to virtually all plant species. To begin with, we have the biolistic approach, that is the particle bombardment. So we will just refresh our memory what you people have studied at your graduation level. In particle bombardment, the for the exogenous DNA to enter the nucleus of a host cell, it must overcome the cell wall and plasma membrane. So the barrier is here in this case is the cell wall and the plasma membrane. So whatever the exogenous DNA, you're trying to push it through the into the cell. So in order to enter the nucleus of the host cell, it must overcome the cell wall and the plasma membrane. Particle bombardment is about the acceleration of inert metal microparticles that are carrying DNA into the target cell. And of course, these microparticles must overcome the cell's barrier without causing cell death. So when we are using the microparticles, that is metal microparticles, of course they are inert, but at the same time they should not cause cell death. It is only to modify the cell wall and the plasma membrane so that the barrier is not there, and the DNA can easily enter into the nucleus of the host cell. So what is done? Initially the exogenous DNA must be incorporated into bacterial plasmids, and in addition to the exogenous DNA of interest, that is gene of interest, the vector should contain a promoter, a terminator, and selectable marker genes for the purpose of tracking the transformation and selection. Right? So for the exogenous DNA to be incorporated, first is it has to be incorporated into bacterial plasmids, and the vectors should contain a promoter, a terminator, and a marker genes or a selectable marker genes for the purpose of tracking the transformation and selection. A high concentration of the DNA vector is used when attaching the vector to the microparticles. In this step, what happens? The carrier DNA, usually the salmon sperm DNA is used. So these carrier DNA can also be added to increase the transformation efficiency. So this is what we have to remember. Something called as carrier DNA is added usually from the salmon sperm DNA for the only reason that to increase the transformation efficiency. So this complex of vector, carrier DNA and metal microparticles must be accelerated to reach inside the recipient plant cell nucleus. So put together, the complex must be accelerated so that it reaches the nucleus of the plant cells. Right. So most of the systems used for accelerating the metal microparticles, they are based on generating great power so as to displace a membrane that contains the DNA coated microparticles. Right. So the systems which are used for accelerating the metal microparticles, they are based on the ability to generate great power so that they can displace a membrane that contains the DNA coated microparticles. Speeds of over 1500 kilometers per hour are often reached. So that is the speed with which the complex, the vector, the carrier DNA and the metal microparticles enter into the nucleus of the host cell. Right. Now the energy that can be generated is from various sources. Number one, a chemical explosion, that is dry gunpowder. Number two, a helium gas discharge under high pressure. Number three, the evaporation of a drop of water through an electrical discharge with either high voltage and low capacitance or low voltage and high capacitance. And number four, the discharge of compressed air. So all particle acceleration processes, they are carried out under vacuum to minimize the deceleration. So the energy generated can be generated from various sources. Number one, a chemical explosion by the use of dry gunpowder. Number two, a helium gas discharge under high pressure. Number three, the evaporation of a drop of water through an electrical discharge with either high voltage and low capacitance or low voltage and high capacitance. And number four, the discharge of compressed air. So what you have to remember over here is all particle acceleration processes, they are carried out under a vacuum to minimize the process of deceleration. Is that clear? So you have to be careful enough to minimize the deceleration when you are working on particle acceleration processes. So it has to be carried out only under vacuum, right? So high density microparticles, say for example gold and tungsten, they are most commonly used in the transformation process. So these particles, they measure about zero point 0.2 to 3 micrometers in diameter, and they should be chemically inert so as to avoid damage to the exogenous DNA or cellular components of the host cell. Right? They are, or they should be inert so that they do not cause any damage to the exogenous DNA as well as to the cellular components of the host cell. Right. So, however, irreversible agglomeration of gold nanoparticles in aqueous solution, this can hinder the introduction of exogenous DNA. So, this is the only disadvantage that is associated with this type of microparticle transformation. There is a possibility of irreversible agglomeration of gold microparticles. So tungsten microparticles has also a limitation. It can oxidize with time, which may harm the recipient cell and obstruct the addition of exogenous DNA. No doubt, high density microparticles such as gold and tungstens are commonly used in the process of transformation, but the limitation is irreversible agglomeration of gold microparticles in aqueous solution. So this can hinder the process of introduction of exogenous DNA, and even tungsten microparticles, they oxidize with time. So this also cause obstruction for the addition of exogenous DNA, and also they may induce or they may harm the recipient cell or the host cell, right? And upon the microparticles entry into a cell, the attached exogenous DNA dissociates by interaction with the cellular fluids of the host cell, and it is randomly integrated into the recipient cell gene. And there are various types of plant tissues, say for example, callus, cotyledon, leaf disc, embryos, and cell suspension that have been used in microparticle biolistic method or bombardment method. So the procedures for the selection and regeneration of transformed cells are essentially the same as those which we have described for genetic transformation using agrobacterium tumefaciens, which we will not be discussing over and again here, because the procedure for selection and regeneration of transformed cells are the same, right? So what happens upon the microparticles entry into the cell? The attached exogenous DNA that dissociates by interaction with cellular fluids, and it is randomly integrated into the recipient cell genome. And various types of plant tissues such as callus, cotyledons, embryos, and cell suspension, they have been used in microparticle biolistics or bombardment methods. Right. So moving on further, we have the protoplast electroporation. Now, what is protoplast electroporation? Protoplast electroporation is used to introduce exogenous macromolecules, that is the DNA, such as the gene of interest, what we refer to as, into the plant cells by reversibly changing the permeability of the plasma membrane. So, as I said earlier, the direct methods is about, or it is dependent upon making changes, the physical and chemical changes to the cell wall and membrane. So protoplast electroporation is also about the same thing, making changes. But here it is making a reversible change in the permeability of the plasma membrane. So this is about the reversible changes with respect to permeability, so that the exogenous macromolecules such as DNA or the gene of interest can gain access into the cell through the plasma membrane. And the protoplasts, they are plant cells that have been stripped of their cell walls through the action of pectinases and cellulases. So the protoplasts, they are the plant cells, but they have been stripped of their cell walls, so they lack the cell wall through the action of the pectinases and cellulases. So as described for the other plant transformation techniques, it is necessary to obtain a vector which contain the exogenous DNA or the gene of interest and a selectable marker gene. Right? To introduce the exogenous DNA into the recipient's cell interior, the purified protoplast, they are mixed with the vectors and the carrier DNA. Again, a complex is formed, that is the protoplast are mixed with the vectors which carries the exogenous DNA along with a selectable marker gene, and this mixture is then exposed to short pulses of continuous current with high voltage. Right. Now what happens? Pores form temporarily in the plasma membrane. A transient pore formation occurs in the plasma membrane. Now you have the mixture of the protoplast, the vectors and the carrier DNA, and now this mixture is exposed to short pulses of a continuous current with high voltage. The pores form temporarily in the plasma membrane, thus allowing the vector carrying the gene of interest to enter into. Right. So by increasing the intensity or duration of the pulses, it is possible to increase the number and size of the pores formed. So this is important. To increase the size of the pore as well as to increase the number of pores, you can manipulate this by increasing the intensity of the pulse of a continuous current with high voltage, or duration of the pulse. So by manipulating the intensity and duration of the pulse, it is possible to increase the number as well as the size of the pores that is being formed. Right? So the voltage and capacity, or sorry, capacitance intensities, the pulse duration, the electroporation buffer composition, and the concentrations of the protoplast and DNA, that is with respect to both vector and carrier, should be optimized to achieve a successful transformation of protoplast by electroporation. So all these are very, very important. Number one, the voltage and capacitance intensities, the pulse duration, the electroporation buffer composition, and the concentrations of the protoplast and DNA, that is with respect to both vector and carrier, should be optimized to achieve a successful transformation of protoplast by electroporation. Is that clear? The major difficulty with this electroporation technique is the regeneration of plants from the transformed protoplast. Plants regenerated from protoplast may also have fertility problems, right? So the major difficulty with this technique or the protoplast electroporation technique is about the regeneration of plants from the transformed protoplast. That is, plants regenerated from protoplast may also have fertility problems. So, if when creating the protoplast, the enzymatic treatment is replaced by a partial plasmolysis of the cells immediately before electroporation, so that these problems with protoplast regeneration can partially be avoided. So, how to overcome the difficulty, at least to some extent, partially to avoid the problem. The problem is about the fertility. So, when creating the protoplast, the enzymatic treatment is replaced by a partial plasmolysis. So, that is before electroporation. So, these problems with protoplast regeneration can partially be avoided. Is that clear? And there is an alternative to electroporation. We have the PEG, that is polyethylene glycol. So these polyethylene glycol associated with calcium and magnesium at an alkaline pH can be used to promote and binding of the gene of interest or the exogenous DNA of interest into the protoplast. So if electroporation is not desirable, then you can have an alternate to the electroporation, that is polyethylene glycol, the PEG we refer to as PEG mediated DNA transformation. And this PEG associated with calcium and magnesium at an alkaline pH can be used for or to promote the binding of exogenous DNA and to get transformed into protoplast. So in this method what happens is DNA adheres to the cell surface and is absorbed by the process of endocytosis. So here you can see the endocytic behavior. The DNA adheres to the cell's surface and then finally it is absorbed by the process of endocytosis. And then the treatment with PEG can also affect the regeneration of plants from protoplast, increasing the difficulty of obtaining transformed plants. So there is some there is a limitation. It can affect the regeneration of plants. So no doubt you can push into the gene of interest into the protoplast into the through the protoplast, but the problem is regeneration of plants from the protoplast. That is another important limitation. So this increases the difficulty of obtaining the transformed plants. So even if you push into the protoplast, the regeneration of plants from protoplast is a challenging task. So that increases the difficulty of obtaining the transformed plants. So with this, we have discussed in detail about the creation of transgenic plants, and we have seen different approaches. To begin with, it was about the agrobacterium tumefaciens, and then we discussed about the direct methods, the biolistic approach, and also about the electroporation of the protoplast And if electroporation is not desirable, we have alternate methods or approaches to electroporation, that is through polyethylene glycol mediated transformation of the DNA. And each of these methods have their own limitations, and there are possibilities that to overcome these limitations. But most commonly used method is the use of agrobacterium tumefaciens for transformation of the gene of interest or the DNA, and ultimately regenerating and creation of the transgenic plant. So with this, I'll stop my discussion over here. We can continue our discussion with a new concept in the next session. Thank you very much for your kind listening. Thank you.